Welcome to Broad Eye, the podcast that explores knowledge gaps in ophthalmology and eye care. Hello, everyone, and welcome to another episode of the Broad Eye podcast. My name is Sean Maloney, and today I am going to be speaking with Dr. Brian Ballios. Dr. Ballios has a number of titles, but I'll let you introduce yourself there. If you don't want, can I call you Brian? Is that okay?
Brian's just fine.
There you
Thanks,
go,
Sean.
Brian.
I'm excited to be here.
Can you tell me what your titles are? You have a few of them.
I wear a few different hats, but I think basically I'm assistant professor in the Department of Ophthalmology and Vision Sciences at the University of Toronto, and I'm the J. Ardith Hill Fighting Blindness Canada Professor in Ocular Genetics Research. And as the name implies, I'm a clinician scientist, and I have my laboratory in the Donald K. Johnson Eye Institute at University Health Network in downtown Toronto, where we're doing exciting work on stem cell therapies for inherited disease. And I'm also a physician as part of my time, and I have a practice at University Health Network and also at Sunnybrook Hospital. So I wear a few different hats, but I'm excited to be at the interface of science and clinical care.
Fair. And just for people listening, you were a very highly recommended guest from one of our other guests, Dr. Alan Slomovic. He says, you need to talk to Brian. Brian is amazing. This guy is going to make waves in stem cell research. So I'll put you on the spot here about stem cell research. So you're going to be the guy making waves.
Dr. Slomovic's really kind. I've known him for many years, and he's doing a lot of great work in the cornea with clinical application of corneal stem cells. But I'm in a different part of the eye. I'm at the back of the eye, as he likes to say, and working on the retina. So a different target.
Yeah, fair enough. Can you talk a little bit about stem cell therapies for I guess in this case, inherited retinal diseases is your focus, and what's the clinical objective with some of the stem cell therapies? Where do these cells come from, et cetera?
Sure. So, patients ask this all the time because people have heard about stem cells in popular media, or they've heard about it perhaps in other contexts, or their doctors have mentioned it. The goal of stem cell therapy for inherited retinal disease is to really regenerate the retina. The idea of how to regenerate the retina using stem cells could fall into a couple of different categories. One of the ways that stem cell researchers first thought about how stem cells would help would be actually to replace the cells that are lost slowly over time in many inherited retinal conditions. And again, I emphasize it slowly over time because these are, in many cases, slowly progressing conditions over many years and decades of life. And the original goal of stem cell therapy, I think, was really to say, well, if those cells are going away over time, can we replace them? And by the cells going away, I mean the light sensitive cells inside the eye. These are called the photoreceptors, the rods and cones, the ones responsible for nighttime vision, the rods, and the cones responsible for daytime vision, as well as their supporting cells. And so cell replacement therapy has for a long time been the major goal of stem cell therapy for these conditions. But I think more and more researchers are starting to recognize that stem cells may have other actions that we didn't anticipate. And one of those is called a neurotrophic effect. Neurotrophic refers to the idea that these stem cells can release chemical compounds, factors. They can produce them like little factories and release them into their environment. And these factors that they release, these chemical compounds, may actually have a beneficial effect on neighboring cells or cells nearby. And that transplanted stem cells may actually improve the health of the cells already present inside the eye. And so maybe not beyond just replacing cells, they may have this effect of improving the health of the retina. It's called neurotrophic effect. And then as time went on and researchers with backgrounds in retinal biology and physicians and clinicians with interest in retinal disease got together and started using stem cells and thinking about how they could be developed into therapies, we realized that some stem cells may actually be able to model disease in a plastic petri dish quite well. And our stem cell models of inherited retinal disease have become more and more sophisticated over time. So sophisticated, in fact, that we can now create tiny, what we call organoids, mini organs, little retinal structures, three-dimensional structures in the plastic petri dish. And we can create these structures with the same genetic changes that occur in the patient, and try to understand, maybe we can learn more about the disease by being able to manipulate these in the plastic petri dish, try new drugs, take away factors, add factors, and develop new therapies. So when I think of stem cell therapy, I think of it in a very broad sense. I think of it not just replacing cells, but I think of all these other things that stem cells could potentially do to help us eventually treat inherited retinal disease.
No, and that's a nice, certainly a nice overview. I like where the research side of things has gone, because I've been out of research for a while now, and didn't have the luxury of working with these retinal organoids in culture. But it would have been a lot of I don't say a lot of fun, but I mean, it must be very informative tools for studying. Now these retinal organoids, you're getting, like, where are these cells coming from that you're using, and then also the cells that you're using in, not necessarily you, but I mean you and anybody else involved in these clinical trials. What are the sources of these cells, of these stem cells, I guess?
Yeah. So just taking a step back to touch on something you said, I mean, it is really exciting that we can now make these little organoids in the dish. When I was doing my PhD studies, and we didn't have retinal organoids, we were trying to grow photoreceptors, light sensitive cells, create them from stem cells in the plastic petri dish, and on a flat petri dish. And the eye is a three-dimensional structure. The retina is a three-dimensional structure. It has length, it has width, it has depth. And in a plastic petri dish, everything grows on a flat surface in 2 dimensions. And we would have these cells growing, and we knew that they were expressing the right proteins that photoreceptors should. But one of the biggest criticisms we always faced was they never took on the proper shape. They didn't look like the photoreceptors look like in the human eye. And now we can actually make these organoids that have, like I said, width and depth and length, and they're three-dimensional structures that self-organize. We can get these stem cells that we're using to model disease and think about cell replacement from a number of different sources. One of the most promising sources are called pluripotent stem cells, and pluripotent refers to the fact that in their native state, these stem cells could potentially make any tissue in the body. They could make nerve tissue, they can make skin tissue or muscle tissue or gut tissue. But using combinations of chemical factors, we can coax these pluripotent stem cells to turn specifically into retinal cells. And we've become quite good at turning these pluripotent cells into retinal stem cells. These are cells that only make the stem cells that only make the different types of light sensitive cells inside the eye. And these are the cells that we are working with now. Pluripotent stem cells is a broad term. One of the oldest sources of pluripotent stem cells is something called the embryonic stem cell. These are cells that were originally isolated from the blastocyst, the earliest stage of human development, and then they're grown in culture now in the dish. And there's many different cell lines. And one of the characteristics of a stem cell is that one of the cardinal properties is that it can self-renew forever. It can just keep dividing and making more copies of itself. And so in labs all around the world, these stem cells, these embryonic stem cells, continue to proliferate and divide, make more copies of themselves. And we take a few every now and then, and we transform them into retinal cells to study them or to try to develop therapies. Embryonic stem cells are one source, but in the last probably 15 years or so, a new exciting source called induced pluripotent stem cells have arisen. And they have the same properties of embryonic stem cells, but we don't need to go to the embryo, the human embryo, to get them. We can get them directly from patients. We can get them from adult patients. Originally, it was from a skin sample, but now we can get them from a simple blood sample like you would give for your routine blood work with your doctor. And from those blood cells, we can transform those cells into cells that behave, for all intents and purposes, just like embryonic stem cells. And now many groups are working on the retinal cells made from these induced pluripotent stem cells, or we call them iPS cells. using the retinal cells that come from those to create these cell therapies or these neurotrophic therapies or these models of disease. Those are probably the embryonic stem cells, and iPS cells have been two of the most heavily studied cell types. We also know that the eye, as it develops from the embryo, actually develops from stem cells itself. And these stem cells are quiescent in the adult eye. That means they don't divide anymore. But you can take these cells out of the eye from the adult and actually get them to grow again in the dish. These are adult stem cells, and we've also worked a lot with those to understand how they behave.
Oh, that's nice. I remember. So you and I have some overlapping experience living and working in Boston, and I've gotten to know a lot of the same people,
Yes.
albeit at different periods. I think I'm older than you, but back then I remember using the these mouse retinal progenitor cells and studying these quite a bit. And about that time, that was 2007, I think, when I was in Boston. That it was just then they're starting to work with the human retinal progenitor cells. So it's pretty exciting times back then. But here we are 14 years later, and you're doing cooler things. But
well, I mean, that work was fundamental. I mean, the work you did in Boston at Skeepin's Eye Research Institute, or with Michael Young and that group, those retinal progenitor cells, the studies of those have actually advanced to the point that there are 2 clinical trials going on right now for using those cells in humans, in patients with retinitis pigmentosa, to try to replace and support the health of the cells in RP, in patients with RP. And I've been involved in one of those studies, and it's a really exciting time. And I think that this is just the tip of the iceberg for stem cell based therapies in the eye. Those studies are using human progenitors, retinal progenitors that come from the developing retina. But many more studies are planned using these cells that come from organoids and are grown from these pluripotent stem cells. So I think we're already in humans with these studies, but I think we're just seeing the tip of the iceberg in terms of what stem cells can do. It's a field that's younger than gene therapy, but it's growing at an exciting rate.
And that's the thing, right? Like you're in the research side of things, or as a clinician, and you see how fast the field is progressing. You literally cannot keep up. It's impossible to read all of the relevant studies that are out there. There's just so much happening. But from a patient's perspective, it's kind of binary. It's like, okay, hey, there's this new field of stem cell therapies, 20 years ago or whatever, how many years ago. And it's like, okay, hey, I'm still blind and it's still not helping me. Right. But there's been 1,000 little steps along the way. And there's certainly been some challenges as well.
There have. I mean, I borrow the stem cell term. I mean, the advances are proliferating. They're growing they're growing off each other. They're changing and evolving as they divide, and they become more variegated, and people learn more about the different potentials of these therapies. But I don't think it's a one size fit all. I don't think that stem cells are a cure-all for all retinal disease. And when I talk to patients with inherited retinal disease, and we sit together and look at their clinical results, we pay special attention to, look, how are things for you on this day? Not thinking about the disease as a lump, but thinking as, wow, look at all of these light sensitive cells you do have. These are cells that might be amenable to things like gene therapy or factors to protect them. And then thinking about patients where maybe they've lost a majority of their cells, and saying, well, gene therapy is maybe not as helpful at this stage. Maybe trying to replace cells is more helpful. And so I think in the end, these therapies are going to be tailored, not just, to a big catch-all disease like RP, but it's going to depend on the stage and where patients are at in their condition. And I think that's exciting, because what it really means in the end, and what we all want as practitioners and also as patients, of course, is choice. People want choice, and they want to have an opportunity to choose options of therapy. And that's really exciting in this field, because there really haven't been many or any options for decades. We've known a lot about the disease, but haven't been able to really talk about options for treatment. And now we can actually talk about that. And that's really cool and very exciting.
Well, and I think it's important, like you mentioned, that there's going to be, different therapies potentially for different stages of the disease. So if you have lost most of your photoreceptors, but still have enough, remaining retinal architecture, well, then maybe you could benefit from, a stem cell based therapy. But gene therapy, at least targeted at photoreceptors, is not really going to help you because you don't have many photoreceptors, right? So it's going to be stage specific. I think a lot of these treatments, and just the fact that a lot of them are evolving very quickly and advancing quickly in parallel is nice, right? That it's not necessarily just going to be, someone who's end stage disease could benefit, or someone who's early stage.
Yeah, 6 or 7 years ago, we had one gene therapy that was just emerging on out of the phase 3 clinical trials, Luxturna, Veretagene, Naparvovec, which is the gene therapy for patients with changes in the RPE65 gene, and now we have tens of ongoing trials. We have for many different genes, for X-linked RP, for X-linked retinoschisis, for genes associated with various genes associated with Leber's congenital amaurosis, a variety of different RP genes. And again, these genetic therapies are proliferating, and several of these are in phase 3 studies. It's quite advanced studies, and that's really exciting. I think we're going to see some more choices emerge, and we're going to start to cover more greater and greater populations of patients who can benefit. Because we have to remember that one of the limitations of gene therapy is that it's an approach that depends in many times, not all cases, but in many cases, depends on treating a specific gene. And we know that there's over 250, almost 300 genes involved in various types of inherited retinal disease. And so, the trials right now are trying to treat the most common, but we don't want to leave anyone behind in the end for these conditions. And I think one of the exciting things about stem cell therapies, or potentials of stem cell therapies, have been true for a long time, is that the hope is that it can treat patients regardless of what gene has been changed. And I think that's been the idea for a long time. Though I'll be honest with you, I think there is evidence emerging that the certain stem cells may not have the same efficacy in all different all different types of inherited retinal disease, depending on the gene. So that's kind of an active area of work where we're trying to understand, maybe stem cells are more helpful with patients with a certain type of gene mutation and not others, and maybe we need to use a different stem cell for those patients. So that's where the field is kind of growing into right now.
But you bring up a good point, because when you're looking from the outside in, and you think this is like just this continuous borrowing from Jim Collins, like the 20-mile march, right? Every day just marching forward, we're making advancements. There are a lot of things that zigzag and even take steps back in the overall progression of anything, certainly in medical research. What are some of the challenges? Anything that comes to mind that the field has faced, whether it's gene therapy or stem cell therapy? Anything that's come up, challenges or obstacles that maybe were unforeseen beforehand? Anything that comes to mind?
Yeah, I mean, one of the things that immediately comes to mind that touches on both stem cell therapy and gene therapy, and even other what we call neuroprotective strategies to protect the cells that are present within the retina, is one of the biggest challenges has been understanding how we assess clinically effectiveness of these therapies. How do we understand whether these therapies are actually having a beneficial effect in patients, given what we know about how patients perform different visual tests, and also what's relevant to a specific disease. RP is a great example where visual acuity is something, reading vision and reading an ETDRS chart, reading the letters on the board is actually something that changes is not the first thing to change in the disease. I mean, patients with RP typically report changes in their night vision and their side vision long before they report changes in their central vision. There's obviously exceptions to that, but that's a general pattern. And so for trials assessing the effectiveness of therapies for macular degeneration, where the reading vision is the first thing to change, I mean, they really depend on outcomes based on how patients perform on an eye chart. It's not really relevant to conditions or to treatments treating early stage retinitis pigmentosa. We really want to know maybe how these patients are changing in response to therapy with respect to their visual field, and how can we assess that accurately, recognizing that, you know, if you have a patient practice time after time, visit after visit, there's a natural learning effect that they can get better at the field over time, whether they have RP or whether they don't have RP. And so creating new and relevant assessments of clinical efficacy has become really, really important. A great example was in the trial for RPE65 gene therapy for Luxturna. They said, well, maybe we shouldn't be thinking about visual acuity. Let's actually test how these patients navigate a visual environment in something that's akin to what they might encounter in real life. So they set up an obstacle course, and they had patients navigate the obstacle course under different levels of light. And they set up pieces of obstacles that would be akin to home furniture, different heights, and tried to adjust levels of illuminance at different parts of the obstacle course, and actually found that this was the test that best measured efficacy with respect to the gene therapy and best captured what patients were telling us. And that brings us to another thing. You know, we depend so much on objective metrics of effectiveness, what we can measure in the clinic. One of the really exciting areas of research is what we call patient-reported outcomes. What are patients really telling us about how their vision is changing in response to these therapies? I've heard many stories from patients after receiving different types of gene therapies talking about phenomena which changes in their color vision, their contrast sensitivity, their sensitivity to bright or dim lights that are very difficult to quantify, or that in ophthalmology we haven't yet developed tools to quantify these. And yet these are real patient-reported outcomes, and we're getting better at capturing that data and using it to inform how we go forward. So that's been a challenge, and those are some of the ways we're trying to get around it. There's other challenges that are cell therapy specific we could talk about, but I think that from a clinical perspective, clinical assessment of outcomes is a big one.
Well, you know, it's really interesting you're talking about that. And I mean, I didn't know the conversation was going to go that way, but I have so many things in my own life that I going through my mind as you're saying these things, right? Like you know, just for the record, my right eye visual acuity is basically light perception, and maybe no more light perception, maybe hand motion on a good day. And my left eye on a good day would be 2400, which also means in a high contrast telephone with, you know, a lot of zoom on, I can still see a few letters at a time. Mm-hmm. But so if I could go from you know, seeing a little bit better there, it might not help my day-to-day that much. But if I could, you know, not, you know, trip over my dogs in the house, or not, you know, bump into people as I'm walking through aisleways and stuff like that, that, you know, for me is more impactful in my life than maybe the improvement in visual acuity. So I think having the patient perspective is certainly useful. But you alluded to some cell-specific challenges. Maybe can you just touch on those a little bit?
Sure. I mean, cell survival and the way in which we can encourage cells to survive after transplantation into the eye and in cell therapy has been a major challenge. You know, cells. What we've learned is that cells on their own undergo a process of programmed cell death called apoptosis, and they, unless they're able to integrate into a tissue efficiently and in a timely fashion, they tend to degenerate and die. And one problem myself and many others have been working on for a long time is how to improve the survival of these cells long enough for them to have their beneficial effect after they've been used in a cell therapy, and whether it be for neurotrophic or it be to actually integrate into the retina itself. And we tried a few different approaches for this, and groups have tried, do we mix the cells together with growth factors and survival factors to help them? One approach that I've taken for a long time is to try to use my experience in biomaterial engineering to create a delivery matrix that these cells can live in, that when they're delivered into the eye and it's an injectable gel, that they can actually survive and live long enough inside the retina to grow and survive and integrate. But cell survival has been a big problem. In the initial studies, survival was far below 1% of the total transplanted cell population, and now we're getting cell survival levels upwards of 20% to 25%, which is a huge improvement when you think about just how many cells you need to actually provide to the retina and get integration. I don't think anyone expects that we'll ever get 100% survival of cells, and no therapy is 100% efficient. But now we're talking about, as a percentage, we're talking about millions of cells now that can actually survive and potentially integrate. And that's really exciting, because we think that we're going to need millions of cells to replace those inside the retina. So cell survival has been a challenge for a long time. And then the other one I touched on was understanding how these cells behave in different conditions, different diseases. For a long time, researchers treated all RP models as sort of akin and the same and interchangeable. But I think what we recognize now is that cells may have different efficiencies at integrating into the retina depending on what gene is mutated, and that's not something you can just try in patients, right? That's not something you can just design a trial to get at. But now with some of these retinal organoid models and these better models of disease in the dish, I think we can actually approach answering that question and then tailoring the cell therapy to the patient and their specific genetic variant.
So I want to take a little bit of a turn and talk about something I know is close to heart for you. Another area maybe making waves, and it's in pathways of care. When we spoke a few weeks ago for the first time, you brought this up, these pathways of care, and that there are some inefficiencies that exist. When it comes to ophthalmology and eye care in general, can you just talk a little bit about the pathways of care, what maybe inefficiencies exist, and where you see opportunities to either yourself or just collectively make an impact?
Yeah, I think it's very challenging for patients with inherited disease to find a home with a provider for themselves that can understand their very specific condition, because these are usually very specific conditions that depend on a specific gene. To find a home with a provider who understands understands how to assess progression of their condition, how to connect them with the advances that may be relevant, and if they're interested in trials, potentially connecting them with something that's appropriate and specific to their stage of their condition. And I think that this is a field that's expanding, but nothing's ever growing fast enough. Nothing's ever growing fast enough for patients and for providers. And so with a small number of providers with focused training in inherited disease, I think we really have to be reaching out, and we have to find better ways to engage and educate our optometry and ophthalmology colleagues, our other retinal specialists who are specialists in other parts of other diseases of the retina that are not necessarily genetic diseases. Engage and educate them in how to manage patients, how to address their concerns, how to understand when they have a patient who's appropriate to refer on for more advanced testing. And as a community, we need to come together better, because it is a time of growth in this field. For a long time, for decades, as I was alluding to before, inherited retinal disease was really a field of ophthalmology and eye care that it was really about making an accurate diagnosis, but there wasn't too much we could do to help these patients beyond counseling them around how to and work with them on how to adapt their vision to their changing life and life demands. But now with the idea that therapies are here and we're doing them in trials and they're actually getting approved, it's even more important that our colleagues know when is it right to send patients on and when is it right to engage them. And how do we talk to patients about what it means to be as part of a trial or part of a therapy, recognizing the trials are not therapies, trials are experiments in humans. And potentially of great benefit, but understanding a patient's desire to participate. And finally, we live in the age of genomic medicine. We live in an age when finding out what gene is causing your inherited retinal condition has a huge implication, not just on understanding and telling you where do you expect to go in the future, but how can you interface with new therapies. And so pathways to genetic testing is a real passion of mine. I think that we're not doing a good enough job as a community of practitioners at getting patients a genetic diagnosis, at getting patients to know what's the gene that's actually causing their inherited retinal condition. And that requires some advocacy on our part and on the part of patient care organizations. I think that we want to, in our healthcare system here in Canada, we want to have efficient genetic testing that's supported by our public healthcare system. And like so many things in our public healthcare system, it's not always the most efficient pathway to get a patient from a diagnosis like you have an inherited retinal condition to it's caused by this gene. And that lag time between the genetic diagnosis and between the diagnosis of you have an inherited retinal condition and the genetic diagnosis, that lag time can be actually several years for patients, and we need to do a much better job. And that's going to require partnering with our colleagues in clinical genetics more broadly, who think about genetic conditions outside of the eye, across the entire body. And it's also going to depend on us as ophthalmologists to become more informed about genes and their roles in disease. But I think, there's a lot of hope here that we're going to be able to push this forward and get more efficient pathways to effective care for patients. So I'm quite optimistic in that area. We can learn a lot from our colleagues around the world and study how they're doing it and bring a lot of those things home as well. And then we have a lot to teach from our healthcare system, which is unique in the world.
Well, I think that we can point at the healthcare system. I think that if you look back, let's say 20 years ago, and someone, goes to their ophthalmologist or optometrist and they get diagnosed, we talk about retinitis pigmentosa a lot, so we can give that as an example. Okay, by the clinical signs and symptoms, you have retinitis pigmentosa. Unfortunately, there's not much that can be done about it right now. We'll do annual follow-ups. If you have any major change in your vision, come back and see me. I think that was kind of the, how it was handled. But since then, there have been some therapies developed. And in the case of Luxturna for RP65, that is now approved by Health Canada. And stories that I've heard, like they're actually trying to find, I think Novartis is involved in making Luxturna available in Canada, but correct me if I'm wrong. And yes, they were actually like trying to find patients. Like they know there's patients out there. There's patients that, could benefit from this. Yeah. But there are people just have never had genetic testing, so they don't know. And it's like they have some sort of campaign to try and find the patients. Like, hi, we have this therapy now that's approved, but who wants it?
It's true.
It's crazy though. That's crazy.
It's true. I mean, there's companies around the world that have become so eager to find patients for these therapies that they've been developing that the companies are actually sponsoring genetic testing. So it's free to the patient. They don't have to pay out of pocket. The company is sponsoring the testing. And in a healthcare system like Canada, it raises a little bit of a challenge in that how do we ensure that the government retains the sort of the responsibility to continue to provide testing when necessary for patients, but there's so much eagerness to find these patients around the country. The scientist in me when I meet with a patient scientist, to me, I want to know the gene. I want to know for all the patients I see. I want to know the gene. I recognize that patients, they come to these decisions. It can be very complex when deciding whether you want to have genetic testing done, the implications of knowing a gene, how it influences what you know about the family, and potentially family planning for future generations. There's a lot of counseling needs to go around that. The decision to get genetic testing shouldn't be made lightly, but I think that there could be, we need to tell patients that there can be a lot of benefit to knowing the specific gene. And like you said, Sean, times have changed, and like you said, it was very different in the past, where you tell the patient, you have RP, and this is what you might expect, and come back and see me in a year or 2 years, and follow up with your regular eye doctor, and let's see how things go. I think we need to, explain to all of our colleagues that another option should be put forward on the table, which is, by the way, some really exciting things going on in this space, and a lot of being able to engage with those exciting things depends on knowing the gene. And the sooner we know the gene, the sooner we can figure out which of those exciting things might actually be applicable to you as a patient. So at the end, the choice is always the patient's, but they should know they have a choice, and we should be able to provide them with an efficient choice. And that's just about good personalized medicine.
Well, I think that it's the last part of that that's been, some of where the frustration comes about. It's not, for the patients that say, hey, I don't really want to know, I'd just rather kind of live my life. Okay, fine, that's your personal choice. But for the ones who want to know, I think that there's a lot of instances where they'll go just to their frontline eye care providers and say, hey, like, where can I get genetic testing? And the answer is like, I don't really know. Right. And that, like, I've heard this numerous times from people, and just they don't really know where to go. And these are ophthalmologists and optometrists. So I think that there's a huge education component out there, again, because maybe when they were going through their training, there weren't these therapies really in the pipeline. So it was just the idea of sending patients for genetic testing was never really part of the follow the care process. But I think moving forward, that it seems to be necessary for patients who want to know what the underlying mutation is, so that they could qualify for therapies that are up and coming, right?
And not every care provider is necessarily going to feel comfortable ordering genetic testing themselves on their patient. I mean, the genetic test, the interpretation of the results of genetic testing can be complex, and can involve a large amount of analysis that has to go into determining whether a misspelling in a gene that a genetic test identifies is actually relevant to the condition that we see in the patient. And so if physicians or their patients are saying, I want to get genetic testing, but they're not sure they're necessarily comfortable themselves, they should know they have resources available, or people to refer to, or to colleagues to talk to who have more comfort in this space and can help navigate that path. So and the other thing that happens is sometimes patients get genetic testing and it doesn't yield a result right away. It doesn't come back, and about 30% of the time, you get a genetic test and it doesn't show right away what the misspelling is in the gene. And patients can get discouraged because they say, well, why did I do that genetic testing and wait that time, and I didn't get it, they didn't find the gene. And part of it is because we don't know all the misspellings in genes that can cause disease. Sometimes we see a misspelling, we don't think it's actually causative of the disease, but it might be. And if it's been a few years, patients have had genetic testing that was negative, I like to have a discussion with patients about how we might go back to the table and try again for genetic testing, because this is a field that's the field of genetic testing and finding these gene mutations. That's also something that's evolving at a lightning pace. And so re-engaging patients who maybe weren't successful the first time through at finding their gene, and reminding them that, look, things have changed incredibly quickly in the last 5-10 years. Genetic testing has been around for a couple of decades, but the efficiency of doing it is improving all the time. So we should be not just engaging new patients with their choices, but we should not be forgetting about those patients who've had some investigations done and never received as clear a diagnosis, and bring them back to the table.
No, I think that's good advice. So, Brian, I've taken a lot of your time, but I want to take the opportunity here to thank you for joining and coming on the podcast. Maybe as you get settled into your various new roles, wearing your new hats, and make those waves that have been promised, then we can get you back in the podcast to share some of the research that's been done by your group and related groups, and share that with the audience.
That'd be a lot of fun. It was great chatting with you, Sean.
Great. Thanks so much. And that concludes today's episode of the Broad Eye Podcast. If you enjoyed this episode, please be sure to subscribe on your favorite podcast platform. Of course, ratings and reviews are always welcome. And you can certainly share this episode with any of your colleagues or friends who might enjoy it. Thanks for listening.